Hello and welcome to my channel. Today we are going to learn about biosynthesis of proline. But before going into the steps, let's first see what type of amino acid proline is. Proline is a non-polar aliphatic amino acid. And if you look at the structure of proline, it is having a pyrrolidine ring. And this pyrrolidine ring is a 5-membered ring. We have an amino group in the cycle, and this amino group here it is secondary amine, whereas in the rest of the amino acid that is primary amine. We have a carboxylic group as well. Proline is a non-essential amino acid. That means our body needs to synthesize it. And for the synthesis of proline, we need a precursor. And that precursor is L-glutamate. Proline is synthesized from L-glutamate. Now, L-glutamate can also be used for the synthesis of other amino acid. We will discuss that in a minute. But if you look at the structure of proline, basically it's a cyclized derivative of glutamate. Now, the proline is also a proteinogenic. It is used in the biosynthesis of the protein. And in the biological condition, basically proline is present as a Zwitter ionic form. Now you might have learned that Zwitter ion, the amino acid is in the ionic form, and here you can see that nitrogen is protonated with one more proton and it is having a positive charge, and the carboxylic group is in the form of carboxylate ion. Now, as I mentioned earlier, that proline we are getting from glutamate, but glutamate is also the precursor for glutamine and arginine amino acid synthesis. Glutamate precursor is alpha ketoglutarate, and alpha ketoglutarate we are getting from the Krebs cycle. So you can see the significance of Krebs cycles. Now, if you look at the structure of glutamate and proline, as I mentioned earlier, that proline is the cyclized derivative of glutamate. If I connect the glutamate from the carbon and the amino group and I cyclize it, then I will get the proline structure, and the carboxylate group, which is here, you can see it is present on the side of the ring. Let's see the biosynthesis of proline. Glutamate will take the phosphate group from ATP. So ATP is changing into ADP. This process is taking place in the presence of enzyme glutamate kinase. The phosphate group will attach onto the carboxylate group, but there are two carboxylate group. So basically it is going onto the alpha, beta and gamma carboxylate group. That is why the name of the product is gamma glutamyl phosphate. In the next step, gamma glutamyl phosphate will releases the phosphate group, as well as you can see that carboxyl group is changing into the aldehyde group. This process is a reduction process. Because it's a reduction process, so we need a reducing agent, and here the reducing agent is NADH or NADPH, along with one more proton is changing into NAD plus or NADP plus. This process is a oxidation process. So we are having both reduction and oxidation going on, and the enzyme is gamma glutamyl phosphate reductase. The product is glutamate gamma semi-aldehyde. So we have one side aldehyde group, one side we have a carboxyl group. Now down I have written in a folded form, as you can see that there are 3 carbon, blue color, there is an aldehyde group, there is a carboxyl group, there is amino group, just as this folded. This molecule, the dehydration will take place. It's a non-enzymatic reaction. Oxygen from the aldehyde, 2 hydrogen from the nitrogen will go out in the form of water, resulting in the cyclization and giving you the molecule pyrroline 5 carboxylase, also known as P5C. Now it's a cyclized molecule, but as you can see here, it's having a double bond, but the proline is not having a double bond. So we need to do a reduction here. That is done by pyrroline carboxylate reductase enzyme, and the 2 hydrogens are added, one on the carbon and one on the nitrogen. For this reduction process, we need a reducing agent, and again here we have to use either NADH or NADPH. that will get oxidized, giving you the reduction process where on the double bond hydrogens will be added. So this is the structure of proline. After adding of two hydrogen, reduction process has taken place. Enzyme, as I already mentioned, is the pyroline carboxylate reductase. Now here I am drawing the dash line formula for the proline as well, so that you know that there is no doubt between the structures. And as you can see, the nitrogen here is protonated and it's having a positive charge. And on the next carbon, next to nitrogen on the carbon, we have a carboxylate group. So the proline is now synthesized by using the different enzymes. And if you look at the overall process, here is the overall process starting from the glutamate to gamma glutamyl phosphate to glutamate gamma semialdehyde to pyroline 5-carboxylase and then to proline. I hope you understand the biosynthesis of proline, and if you like this video, please make sure to hit the like button, share and subscribe to my channel. If you want me to make video on any specific topic in chemistry, organic chemistry or biochemistry, feel free to mention it in the comment section. Thank you for watching and see you in the next video.